Imagine suffering from a severe medical condition, say, rapid hearing loss.
That sounds terrifying.
Right. And now imagine finding out that the exact biological mechanism to prevent your hearing loss was actually discovered and published 10 entire years ago.
Wow.
Yeah, it was just sitting in a public database the entire time. But nobody realized it simply because the scientist who understood the ear had never read the paper published by the scientist who was studying cancer. The cure was there, completely ignored, just trapped in a PDF.
Which is, sadly, the daily reality of modern science. I mean, we often assume that breakthroughs are just waiting on some massive new experiment, right?
Like we just don't have the data yet.
Exactly. But the truth is, the human brain has a strict bandwidth limit. Humanity is publishing papers and generating raw data vastly faster than human minds can cross-reference it all. The bottleneck is no longer a lack of information, it is lack of synthesis.
Which brings us to the focus of today's deep dive. Whether you are a researcher trying to stay afloat in this sea of literature, or a business strategist trying to connect isolated data silos in your industry, or just someone who hates information overload, this really matters to you.
It definitely does.
Because today we are looking at a May 2026 paper from the journal Nature. It introduces Robin, which is a multi-agent AI system designed to act as a semi-autonomous scientist. And it doesn't just write text. It actually generates hypotheses, designs physical lab experiments, and analyzes raw lab data. Okay, let's unpack this, because Robin is attempting to solve that exact bottleneck of human synthesis you just mentioned.
Right, and to understand the solution, we really have to look closer at the cost of that bottleneck. That hearing loss example you gave is actually very real.
Wait, really? That actually happened?
Oh, absolutely. There is a drug called dabrafenib. By 2010, the scientific community had thoroughly mapped its molecular action. They knew it inhibits something called BRAF kinase, and they were using it to treat certain cancers.
Okay, so they knew how it worked.
They did. But the logic connecting that BRAF inhibition to preventing hearing loss just wasn't put together until 2020.
So the biological breadcrumbs were just sitting there for a full decade.
They were just sitting there. The ear-protecting effects were a completely logical extension of its known mechanism. But, a human mind hadn't read that specific combination of papers to connect the dots.
That is so frustrating.
And when the connection was finally made in 2020, it wasn't even through targeted reading. It was found through unbiased, high-throughput screening.
Which, for anyone who hasn't spent time in a wet lab, is essentially the scientific equivalent of throwing thousands of different chemicals at a wall just to see what sticks.
Right, yeah.
It's automated trial and error, rather than starting with a targeted logical hypothesis.
That is a very fair way to describe it. It's essentially brute-forcing discovery because we just can't synthesize the logic fast enough. And dabrafenib isn't some isolated case. The paper highlights a 22-year lag before researchers realized they could repurpose ketamine for certain treatments.
22 years.
Yeah, and a 13-year lag for a schizophrenia drug called CAR-XT. We are talking about billions of dollars in development costs and years of delayed treatments, all because the insights just didn't crystallize in a human mind.
I mean, I definitely understand the problem. But how does an AI actually do science without just spitting out glorified Wikipedia summaries? Because I can open a chat window right now and ask an AI to summarize a biological pathway. But that is very different from designing an experiment that cures a disease.
Right. The difference really lies in the architecture. Robin isn't a single chatbot. It is a structured system that breaks the scientific method down into discrete, highly specialized tasks. But before we even look at how it works, look at the sheer scale of the cognitive labor it replaces.
Okay, hit me with the numbers.
So the researchers calculated the time on task for human scientists to manually read 551 specialized papers, brainstorm hypotheses from that reading, and then write the code to analyze the resulting lab data. The estimate was 359 to 424 hours of intense human labor.
Oh, wow. That is basically 10 weeks of full-time PhD-level work.
Exactly. Robin completed that entire cycle in less than 2 hours. It synthesized all 551 papers in just 30 minutes.
That is insane.
And the API cost to run that entire cognitive cycle, generating the hypotheses, formulating the experimental design, calling the different AI agents, it was just $10.76.
Okay, wait, I do have to push back on that $10 figure a bit. Because that is just the API inference cost for a single run.
Yes, that's correct.
Right, so that doesn't factor in the millions or really billions of dollars of raw computing power and electricity it took to train the base model in the first place. I mean, it feels a bit like saying a taxi ride only costs $10, but completely ignoring the cost of building the car and paving the roads.
Well, that is a very valid critique. You are looking at the marginal cost of one inference run versus the massive sunk cost of training the underlying models. The underlying infrastructure is profoundly expensive.
Right, somebody had to pay for all those server farms.
Yeah. Absolutely. However, from the perspective of a university lab or a pharmaceutical startup running the system today, that $10 marginal cost represents a total paradigm shift in how they can allocate their budget and human capital.
Okay, that makes sense. The barrier to entry, the cost of access, is what's dropping so dramatically. So you mentioned Robin relies on different AI agents. Let's get into that. Walk me through that structure.
Sure. So Robin divides the labor among three highly specialized language agents. They call them Crow, Falcon, and Finch. First, you have Crow, which basically acts as the scout.
Okay, it's a scout.
Its directive is to conduct rapid, concise literature reviews across hundreds of papers to find initial experimental strategies and therapeutic candidates. It essentially casts a very wide net.
So Crow is the agent doing that 30-minute speed reading session through the 551 papers.
Yes, exactly. Once Crow builds a list of potential drug candidates, it hands them over to Falcon. Falcon is the deep dive analyst.
I like these bird names.
It's very thematic. So Falcon takes each candidate and generates a comprehensive, rigorous report. It evaluates the pharmacology, the scientific rationale, and the potential limitations of every single option.
Okay, so Crow finds the ideas, and Falcon rigorously evaluates them.
Right. And once those reports are generated, an underlying large language model acts as a judge. It runs a tournament to rank these candidates through pairwise comparisons.
Let me make sure I'm visualizing this tournament correctly. Is it like a round-robin sports bracket? Like it pits drug A against drug B, evaluates them, and then puts the winner up against drug C and just tallies the wins?
That is a highly accurate analogy, yeah. Because instead of asking a model to rank 100 items all at once, which AI models are notoriously bad at, they tend to lose track of the items in the middle of a long list.
Right, the lost in the middle phenomenon.
Exactly. So the pairwise comparison forces the model to only evaluate two things at a time. It basically asks based on safety and potency, which is better, A or B, and it just repeats that process hundreds of times until a statistically robust ranking emerges.
That is brilliant. Okay, so Crow finds the candidates, Falcon inspects the details, and the tournament logic ranks the best options. But how does Robin handle the actual lab data once a physical experiment is run? Like, who does the math?
That falls to the third agent, Finch. Finch is the data cruncher. When the human scientists run the physical experiment, they upload the messy, raw, biological data files right back to the system. And Finch operates autonomously inside a Jupyter notebook.
Okay, for those listening who aren't writing code every day, a Jupyter Notebook is basically an interactive digital document. You write a chunk of code, you run it immediately, and you see the resulting graph or statistical output pop up right below it.
Yes, it's a very dynamic way to write code. And critically, Finch doesn't just use hard-coded, copy-pasted templates. It dynamically writes the analytical logic line by line, writing real-time Python and R code based on the specific shape of the messy data it's seeing.
Wow, so it adapts to the data on the fly.
It does. It writes the code to run statistical tests, generate publication-ready plots, and even gate flow-cytometry data.
Hold on, let's clarify gating the data. Because raw biological data is rarely clean, right? Gating is essentially drawing a digital boundary around the specific cells you actually care about. You are filtering out the dead cells and the background debris so you can actually isolate the signal you were looking for.
That is exactly the core function of gating. It's separating the signal from the noise. And Finch does all of this while executing its code inside a Docker container.
Which is basically a secure digital sandbox. Like if the AI hallucinated a command that tried to wipe a hard drive, it would only crash that isolated sandbox, not the lab's actual network.
Right, it provides essential security and stability for the whole system.
Okay, here's where it gets really interesting though. Because we all know that base AI models hallucinate. They confidently make things up.
They certainly do.
And I mean, if an AI hallucinates a fake legal precedent in a brief, it's embarrassing. It's a bad day in court. But if an AI hallucinates a biological mechanism or invents a fake chemical reaction, a lab could end up wasting months of time and millions of dollars physically testing a ghost. So how does Robin avoid confidently lying about science?
The researchers were highly aware of this risk. To address it, they performed an ablation study.
Ablation study?
Yeah, in software engineering, an ablation study involves systematically stripping away parts of a system to see how vital they are to the overall performance. It's basically like pulling a spark plug out of an engine to see how the car runs.
I love that analogy. Okay, so what did they pull out?
They bypassed the specialized Crow and Falcon agents entirely. They just asked a highly capable base model, specifically OpenAI's O4 Mini, to generate the drug candidate proposals all on its own.
Oh boy. I have to assume that without the specialized framework, the base model just hallucinated the results.
It was a massive failure. The base model hallucinated 44.5% of its scientific references.
Wow, wait, almost half?
Nearly half of the published papers it cited to justify its drug proposals literally did not exist. It just completely made them up.
That is basically a coin flip on whether you are chasing a phantom.
But when they reactivated the full Robin architecture, when Crow was forced to meticulously retrieve the context, and Falcon was forced to independently cross-examine it, hallucinations dropped to 0.
0. That is incredible.
The multi-agent cross-checking mechanism completely eliminated the fake references. It literally forces the system to verify its own logic against the retrieved literature before moving forward.
That really emphasizes that the future of this technology isn't just about building a bigger, smarter base model. It's about the workflow and the environment you force the model to operate within.
Absolutely. The guardrails are what make it a scientist and not just a storyteller.
Exactly. So let's look at the real-world test. Because the researchers didn't just leave Robin in a computer simulation, they deployed it against a major human disease, specifically dry, age-related macular degeneration, or DMD.
Which is a very severe condition.
It really is. It is the leading cause of irreversible sight loss in the developed world. There's something like 1.5 million people with vision-threatening DMD in the U.S. alone. And because of the aging population, that number is actually expected to triple by 2050.
What's fascinating here is how Robin approached the pathology of the disease. When tasked with finding a treatment, Robin deployed Crow to review 151 recent papers. It identified 10 potential causal mechanisms for the disease and ultimately proposed an experimental strategy focused on something called RPE phagocytosis.
Let's break that down because I know we have some non-biologists listening. RPE stands for retinal pigment epithelium. And these are basically a layer of cells in the back of your eye that act like a garbage disposal system for your retina, right?
Right. Your photoreceptors, the cells actually detecting light, they are constantly renewing themselves by shedding their outer segments. And the RPE cells have to physically engulf and digest that cellular debris.
And that eating process is what we call phagocytosis.
Exactly. But as we age, these garbage disposal cells get sluggish. The debris builds up into these toxic deposits, which eventually kills the surrounding eye cells and causes the blindness associated with macular degeneration. So Robin's hypothesis was really straightforward. If we can find a drug that makes these RPE cells better at their garbage disposal job, we can clear the toxic deposits.
And Robin actually designed the physical experiment to test this. It proposed using flow-cytometry combined with Frodo beads. I just love this mechanical detail.
It is very clever.
Because these Frodo beads are like microscopic glow sticks. But they only light up when they are exposed to highly acidic environments, like the inside of a cell's lysosome, which is basically the cell's stomach.
Right. So they are dark until they get eaten.
Exactly. So if the eye cell eats the bead glows. And if a drug makes the cells eat more beads, you get a much brighter signal on the data readout.
It's an elegant way to measure it.
So the human scientists took Robin's protocol, they went into the physical lab, pipetted the cells and the drugs, and then fed the raw data files right back to Finch. So in this setup, the humans are essentially just acting as the physical API for the real world.
That is a very apt way to describe it, actually. The authors call this the lab-in-the-loop paradigm. The AI is orchestrating the intellectual discovery cycle, while the humans just provide the physical actuation in the laboratory.
Okay, let's talk about what they actually discovered, because the results are wild. After running the screening tournament, Robin identified a drug called Ripasudil.
Yes, Ripasudil.
Which is a drug currently approved in Japan for glaucoma.
Yeah.
But it had never been proposed for dry macular degeneration before. And when the human hands tested in the lab, it was remarkably effective.
It really was.
It was 713 times more potent at making these primary human eye cells eat the debris than a standard benchmark research chemical.
713 times. That is not a small margin.
No, it's massive. And because Ripasudil is already an approved drug, it has a known human safety profile, which theoretically cuts years off the timeline to get it to patients.
Right. Repurposing drugs skips so many early safety trials.
Then Robin also found another one, right?
Yeah.
A drug called KL001.
KL001, yes. KL001 is a circadian clock modulator. And the authors note that absolutely no one had ever proposed KL001 as an enhancer for this specific cellular eating process.
So how did Robin even think of it?
Well, Robin suggested it because it synthesized isolated literature hinting that phagocytosis in the eye is tightly synced to our natural day and night cycles.
Wow, that alone is an incredible connection to make.
It is. But Robin didn't stop at just finding a drug that worked. It actually proposed a follow-up experiment to figure out the underlying molecular reason why it worked.
Okay, this is where it really acts like a scientist.
This highlights the true iterative capability of the system. After Finch analyzed the flow-cytometry data and confirmed that Ripasudil was massively increasing the cellular eating process, Robin told the human scientists to run an RNA sequencing experiment on the treated cells.
Okay, and RNA sequencing is essentially taking a snapshot of the cell's active blueprints. You want to see exactly which genes are being turned on and which are being turned off in response to the drug.
Perfect analogy. So the human scientists ran the RNA-seq and handed the raw genetic data back to Finch. Finch then dynamically wrote the code to perform a differential gene expression analysis.
Which is essentially like comparing a company's budget before and after a new CEO takes over.
Yeah.
You are looking at the two spreadsheets to see exactly which departments suddenly got double the funding and which ones got their budgets cut.
That is a great way to visualize it. So Finch compared those budgets and found a massive threefold upregulation, a huge budget increase, for a specific gene called ABCA1.
Okay, so why does ABCA1 matter in the context of blindness?
Well, if we connect this to the bigger picture, the ABCA1 gene encodes for a lipid efflux pump. Its job is to pump cholesterol and fats out of the cell.
Okay.
This is critical because ABCA1 directly interacts with a protein called apolipoprotein E, or APOE. And APOE is one of the most significant, well-established genetic risk factors for macular degeneration.
Oh, wow. So Robin didn't just find a drug that cleared the debris, it uncovered a hidden molecular bridge, linking the drug's effect directly to the underlying genetic root cause of the disease.
Exactly. It synthesized two very different biological processes. It connected the cytoskeletal changes to the transcriptional changes.
Okay, let's unpack that jargon because it's important. The cytoskeletal changes are the physical shape-shifting the cell has to do to physically engulf the debris, right?
Yes.
But the transcriptional changes are the new genetic instructions being read. It's basically the difference between a worker physically putting the trash in a bigger bag versus the cell actually calling the dump truck to haul the resulting lipid waste away.
Exactly right.
So Robin connected the local physical action to the system-wide genetic response. I mean, it's not just finding a needle in a haystack. It's realizing that the needle is actually the key to a completely different door that human scientists hadn't opened yet.
And that level of synthesis goes far beyond simple information retrieval. It requires integrating complex biological mechanisms across entirely different domains of literature.
Right. But I'm sure someone listening is wondering about alternative tools. Like, if I have access to OpenAI's new deep research tool, which is specifically designed to conduct multi-step research on complex topics, couldn't I just feed it the same prompt and get the same drug candidates?
That is a very valid question, and the researchers actually asked that exact question themselves. They set up a head-to-head showdown.
Oh, I love a good AI showdown.
They gave the exact same initial prompt to OpenAI's deep research agent. Deep Research spent its time crawling the web and confidently generated 17 unique drug candidates for this disease model.
And I assumed the human scientists went back to the lab and physically tested those 17 candidates.
They meticulously tested every single one on primary human eye cells in the lab, and 0 of them worked.
0. Not a single hit.
Not a single hit. Furthermore, Deep Research completely failed to identify the ROCK inhibition mechanism that Robin so successfully zeroed in on with Ripasudil.
That is wild. Why did Deep Research fail so completely when the underlying intelligence of the models is so similar?
Well, largely because Deep Research operates in a vacuum. It likely hit the limits of its context window trying to hold all the biological complexity at once. But more importantly, it lacked the iterative lab-in-the-loop toolchain.
It couldn't see the results.
Exactly. Deep Research couldn't test a hypothesis, look at the raw flow cytometry data, realize its initial assumption was slightly off, and adjust its strategy. It was just guessing based on text.
This raises an important question about how we evaluate AI progress, doesn't it? Because we hear constant hype about base models getting smarter and parameters scaling up.
This raises an important question, yes. because the disparity is even more obvious when evaluating pure data analysis.
Mm.
The researchers tested Finch against a standard base model on a benchmark called Bixbench.
Bixbench, okay.
Yeah, it tests complex bioinformatics tasks like genomics. A standard base LLM scored exactly 1%.
Just 1%?
Yes, but Finch, operating in its specialized agent harness with access to its Python environment, scored nearly 23% overall and achieved 100% accuracy on the flow-cytometry workflows.
That is a massive difference.
It shows that a massive neural network might contain the vocabulary of biology, but without specialized guardrails and the ability to dynamically test code, it just fails at the actual practice of science.
So what does this all mean? If you're listening to this right now, the real takeaway here is that the era of combinatorial synthesis has arrived.
It really has.
The most valuable discoveries in your field, whether that involves pharmaceutical research, material science, or corporate strategy, they're likely already hiding in the connections between isolated data silos. The insights exist. But no single human has the bandwidth or the cross-disciplinary expertise to read the millions of documents required to connect them. Systems like Robin act as the synthesis engine. They're basically built to harvest the low-hanging fruit that human experts have been just walking past for a decade.
It is a profound shift, but we also do have to acknowledge the safety guardrails here.
Oh, absolutely.
When a system can perfectly synthesize knowledge to create potent biological agents, caution is non-negotiable. Robin was designed with strict parameters. It actively prioritizes drug candidates with established human safety profiles.
Which is good.
And it runs all of its queries through safety classifiers designed to specifically prevent the generation of malicious biological protocols. And most importantly, it relies entirely on the human lab-in-the-loop for validation. The AI cannot automatically inject a patient or synthesize a new chemical on its own.
Right. The human scientists are still the hands holding the pipettes. We are the physical failsafe. But I want to leave you with one final thought to explore on your own. Right now, a system like Robin can perfectly synthesize the entirety of human knowledge to make a scientific discovery for about $11.
Right.
And right now, it relies on human scientists to actually perform the physical labor. But what happens when the robotics industry catches up to the software? When automated liquid handlers and robotic cell culture systems integrate directly with a system like Robin?
That is the big question.
Right. When the lab-in-the-loop no longer requires human hands at all, and an AI can hypothesize, physically test, analyze, and discover 24 hours a day, 7 days a week, are we about to reach a point where science advances faster than human comprehension can even follow?
It is a scenario that is moving from science fiction to practical engineering very, very quickly.
It's definitely something to think about. Thanks for joining us on this deep dive. We'll see you next time.